Thank you very much for the opportunity to talk to you today about the use of monoclonal antibodies for HIV treatment. So in the last several minutes, I will try to highlight where the field is in terms of exploring the use of broadly neutralizing antibodies. So as you all know, great progress has been made against HIV infection, both in treatment and in prevention, where now we have a combination of long-acting ARVs that can be used for treatment, and a long-acting ARV has also been shown to be more effective than daily PrEP for prevention. However, despite this great progress, there are still challenges associated with antiretroviral therapy. So as we know, once ART begins, there is a dramatic drop in viral loads. However, HIV persists in long term in a reservoir of CD4 T cells that harbor replication competent viruses, such that what is expected if ART is to be stopped, as illustrated in this graph, the vast majority of individuals will experience viral rebound where viral loads are going to increase. And even with suppressive ART, HIV is associated with long-term comorbidities. And it's also daily use of ART is associated with pill fatigue, and then individuals that live with HIV still struggle with stigma. And on a global level, ART is associated with high costs. So in view of these ongoing challenges, we ask the question, can antibodies have a role in HIV infection? And so first, taking a step back, antibodies against HIV that are broad and that are neutralizing are generated during HIV infection, despite specific characteristics of HIV that makes broad neutralization challenging. And some of these characteristics are listed here. So first, envelope is sparsely expressed on the surface of the virion. And these proteins or the epitopes to which antibodies can bind, they are masked by a very complex shield of. More importantly, as I mentioned, HIV envelope is highly diverse. And that makes it challenging for a given antibody to have activity against different strains of HIV. However, despite these challenging characteristics, over time, with this interplay between virus and antibody responses, a subset of individuals go on to produce what's called broadly neutralizing antibodies, which are antibodies that have activity against different viral clades. And this is illustrated here. So in the last 10 years, methods to single sort B cells have been developed. And this allowed the identification of different areas on the envelope trimer, which are illustrated here in this picture with the different colors. And now there are antibodies against these areas of the envelope that have entered clinical development. And what is their function? So in this graph here, we are these different antibodies, and each of these numbers in different antibody. And this shows their breadth, so the percentage of strains that these antibodies are expected to cover to have activity against, and their potency. So before these newer methods were available, only a handful of antibodies had been evaluated clinically, and they were far from the ideal antibody, which I tried to illustrate it here with this red star. And these would be antibodies that would be potent at very low concentrations and then have activity against all of HIV strains. Unfortunately, even though we have made a lot of progress, we don't yet have one single antibody that would have all of these features. However, combinations of different antibodies can get us close to this ideal coverage. Now that we have these various tools, these various antibody tools that have become clinical candidates, we have been able to start to assess potential roles of antibodies in HIV infection. And then these can be grouped for this presentation in two, where antibodies are being considered as long-acting alternatives to ART because they are considered safe as a class, and also because they have extended half-lives that can be made even longer with simple modifications. In addition to that, and perhaps more interestingly, antibodies are being considered as part of what's called treatment-free remission. And that is being considered because antibodies, besides being able to clear free circulating viruses, antibodies can also interact with the immune system in a way that they have the potential to clear infected cells or enhance immune responses such that the remaining reservoir can be prevented from causing viremia. But first, let's look at what antibodies have been shown to do in small clinical trials. So first, antibodies were evaluated for their activity against circulating virus. And then here, individuals that were not on ART and have had measurable viruses in their plasma received a single antibody or a combination of antibodies. And then what these graphs are showing is the decline in viremia over time. And what we find across these different studies, and these are just two examples, is that a subset of participants had resistance to the antibodies, despite these antibodies have been found to be very broad. Within an individual living with HIV, there is great diversity of viruses, such that we found that when you come particularly with a single antibody, it's likely that there will be a pre-existing resistance that will be selected for by a single antibody. However, the antibodies that were administered did have direct antiviral activity, leading to these transient declines in viremia. Viral suppression, full viral suppression, was only achieved in individuals that had low starting viral loads. And this was also recently shown and published with another antibody called PGT121, where two participants showed low or undetectable viral loads for a very extended period of time. And as I mentioned, we saw selection of resistant strains when one single antibody was administered. So if antibodies, one or two antibodies, were not across the board successful in achieving full viral suppression and maintaining viral suppression, can we then use antibodies as a transition in a switch regimen in individuals that have achieved viral suppression with combination ART, as illustrated in this study design figure here, where they came into the study and they received a combination of antibodies over a period of time after they discontinued ART. So in these studies, besides asking the question, can antibodies maintain viral suppression, we also try to address potential effects on the reservoir and also on immune responses. So just going back to the beginning of the presentation, I said that in the vast majority of individuals, when ART is discontinued, we expect the viral rebound will occur within 2 to 3 weeks of ART discontinuation. And what do we find when we administered an antibody? So what this Kaplan-Meier curve is showing is the percent of individuals over time that maintain viral suppression in the absence of ART. In black, we are representing what's expected if you're just to discontinue ART, where viral rebound will occur within a few weeks. When we administered 3BNC alone, we are able to maintain viral suppression for about 8 weeks. However, the antibody did exert pressure on the viruses within the reservoir, such that the rebound viruses were typically monogenic, and they harbored resistance to these antibodies, to this one antibody. So even though we maintained suppression for a period of time, it wasn't successful over an extended period of time because of selection of resistance. So next, we asked the question, well, can we improve by giving a second antibody that targets the envelope by a different angle? And then in fact, we were able to maintain viral suppression for a longer period of time, as illustrated here in blue. And then we have more recently pushed this even further to the right when we gave the two antibodies over a course of 20 weeks. In this last study, we were able to achieve long-term viral suppression with the two antibodies alone in 75% or so of the individuals during the dosing period of the study. And in fact, viral rebound only occurred when one of the two antibodies, in this case 3BNC, that has a shorter half-life, fell below what we believe is a therapeutic threshold. So we expect that with a combination of two antibodies in a subset of individuals that harbor sensitive viruses, if we are to continue to maintain antibody levels over a certain threshold, we would be able to maintain virus suppression long-term. In addition to that, and this, I put this here just as illustration, across the two studies that we did with the combination of 3B&C and 1074, there were these 4 individuals, each illustrated by these graphs, where in black, the black line is the viral loads over time, and the colored lines are the levels of antibodies. So in these 4 participants, they were able to continue to maintain viral suppression for an extended period of time for over a year. And in some cases, well beyond the period of time when the antibodies were measurable in serum. But however, what we don't really understand is that if there are common features across these 4 participants that are implicated in this long-term viral suppression that appears to go in some cases beyond the antibody treatment period. So to summarize this part, us and other groups have shown that HIV bNAbs or neutralizing antibodies are generally safe. The naturally occurring antibodies have half-lives of about 2 to 3 weeks. When given in individuals that have ongoing viremia, and single antibodies can lead to transient decline in viremia. A combination of 2 antibodies maintain lower levels of viremia for a longer period of time. And we are able to show that combination of 2 antibodies can maintain virus suppression for an extended period of time in individuals that harbor sensitive viruses to both antibodies. And I didn't have a chance to show here, but as I mentioned, antibodies have been modified to prolong their half-lives, such that it is conceivable that these antibodies could be administered subcutaneously, perhaps every 3 months, or as intravenous infusions once or twice a year. However, as I tried to illustrate it as I went through the various studies, viral reservoir diversity continues to be a challenge when we think about implementing bNAb-based strategies going forward. And this is something that the field is trying to address by developing new antibodies that have more than one specificity. However, while these newer antibodies are being developed, the currently existing antibodies are being evaluated in different contexts, both in prevention, as will be discussed later today, but also in populations that have, in general, have difficulty with daily regimens. And now I will just turn to the potential role of bNAbs in the context for HIV cure or HIV remission. And as I mentioned, antibodies are being considered in this area because in addition to having direct antiviral activity, antibodies have other effector functions through their FC portion, such that they have the potential to interfere with the reservoir. And in doing so, it is possible that the antibodies can limit the establishment of the HIV reservoir if they are administered with early ART. They have the potential to reduce the existing reservoir by directly depleting infected cells, or they may be able to control the reservoir and prevent viral rebound from occurring by enhancing existing immune responses. However, we know that antibody activity depends on binding to HIV, to the antigen. Can we achieve this level of binding and modulation of cellular immune responses in the presence of suppressive ART? This is a question that we still don't have an answer for. And will long-term control require additional immune modulation to the antibodies in the form of vaccines or TLR agonists, like we have seen in cancer therapies? So what do we know so far? We know from a series of studies in non-human primates that are illustrated here that combination immunotherapy, and that is a combination of antibodies with either a vaccine or TLR agonists or a cytokine, were able to lead to long-term suppression in each of these studies in a subset of animals. In all of these studies, immune effects from the immune-modulated agent were observed, either in NK cell function or T cell function. As I mentioned, long-term control was achieved in a subset of individuals. And even though we don't fully understand the mechanisms that were involved in this long-term control, across the studies, there was evidence that CD8 T cells were involved. And in addition, some of these studies show that this long-term control may also be related to the reservoir size or composition in addition to the immunologic features. But what do we know so far in clinical studies? So this is again a cartoon illustrating the different functions of antibodies. And then when we're thinking about antibody effects on the reservoir, we are focusing on their FC mediated functions that can trigger ADCC and clearance of infected cells, or by forming immune complexes, enhance antigen presentation to dendritic cells, and then by doing so, enhance cytotoxic T cells. So in our studies, we evaluated effects on the size and the composition of the reservoir by a viral outgrowth assay. And this is summarized here in these graphs, where participants that were able to maintain viral suppression with the two antibodies, 3BNC and 1074 alone, over a period of 3 months, they had their reservoir assayed by this outgrowth assay twice at baseline, as illustrated by the blue bar, and 3 months later by the gray bar. Unfortunately, in this short-term study with this type of assay, we were not able to see a significant change in reservoir size, even though in some of these participants, we saw a trend towards a smaller reservoir size. In contrast, when we looked at cellular immune responses, and here each graph is showing the percent of GAG-specific CD8 or CD4 T cells over time. And then you see that there is an increase in these types of HIV-specific T cells over time after participants went off ART and transitioned to antibodies and continued to maintain suppression. And these findings here were in contrast to a group of individuals that we did assay in the same way that had maintained suppression on ART. So we were able to show that under antibody-mediated virus suppression, there was an immunologic effect in cellular responses. However, this, for this period of time, wasn't sufficient to impact reservoir size. So to summarize this part, what we know so far from our studies and others is that antibodies can engage the host immune response through FC-mediated functions. We have shown that antibodies can enhance humoral responses, even though I didn't have a chance to go over this here today. We show that they can enhance T cell responses. However, these short-term studies have failed today to show a significant effect on reservoir size. So we are waiting to see what long-term studies with antibodies alone or in combination with other immune modulators will show. And this brings me to this slide, where when we move to HIV remission, there are several studies that are building on from the successes seen in non-human primate studies, where antibodies are going to be given either alone in the context of primary HIV infection or in combination with other immunomodulatory strategies to see if we can impact an effect on the HIV reservoir. So just to conclude, taking a step back, when we look at the use of antibodies in the context of prevention in therapy, and prevention, Mike Cohen is going to cover in greater detail, but now we do have proof of concept that antibody can mediate prevention of infection by antibody-sensitive viruses. We also have emerging evidence that antibodies can maintain viral suppression in individuals that harbor sensitive viruses. Antibodies as a class have potential advantages in contrast to ARVs because of their safety profile and because they would not select for ARV resistance, for example. However, there are challenges that the field still needs to address, which are the issue of pre-existing resistance when we look at circulating viruses in the context of prevention, or within the latent reservoir in the context of HIV therapy or the use of antibodies for HIV remission. Turning over to remission, antibodies are in the center of various studies that are aiming to achieve this long-term virus suppression in the absence of therapy. However, this remains an aspirational goal so far that will likely require combinations, as we saw in the series of non-human primate studies that I showed earlier. Those studies have been very promising, and they have served as the background and have generated supported data that led to the development and launch of a series of clinical studies testing different strategies. So I expect that over the next 2 years, we as a field will have a greater understanding of what antibodies can do in the context of HIV remission by either modifying the reservoir or enhancing immune responses, or a combination of both. And then with that, I will conclude by, the studies that I alluded to were done in collaboration with a large group of collaborators from many institutions. Our studies here at Rockefeller are done in close collaboration with Michelle Nussenzweig's laboratory. And then we are indebted to all of our funders. Thank you very much.